Let's talk about transformation. Transformation is the process by which DNA is introduced to a bacteria. Bacteria can then replicate and express the DNA, which can be isolated for downstream applications. Artificial transformation of bacteria involves producing competent cells capable of exogenous DNA uptake. There are two primary methods of artificial transformation used in laboratory: chemical transformation and electroporation. Chemical transformation requires chemically competent cells, which uses dynamic cations to increase the permeability of the bacterium cell wall, thereby increasing the likelihood of DNA acquisition. Many common protocols, including the heat shock steps, is employed to improve DNA uptake. Electroporation involves the application of an electric field to the cells, thereby introducing holes in the cell walls. The cell wall now allows exogenous DNA, or DNA from outside the organism concerned, to pass onto the cytosol of the bacteria. On the other hand, Griffith first discovered the phenomenon called transformation in 1928. In his experiment, a pathogenic S strain of Pneumococcus forms a smooth colony on Petri dish. Because the bacteria are surrounded by a slippery polysaccharide capsules, a non-pathogenic R strain lacks the capsules and form rough colonies. An R strain can be transformed into an S strain. In this experiment, DNA is extracted from the S strain. The purified S DNA is added to a culture of the R strain bacteria. The bacteria take up fragments of DNA from the environment. The DNA passes through the cell wall and plasma membrane. One strand of the foreign DNA degrades. Recombination of the foreign DNA strand in the bacterial chromosome is accomplished by a pair of crossing over events. The result is a double-stranded DNA in which the two strands are not complementary because a part of one strand came from a different source. The leftover DNA is degraded. Both strands of the DNA replicates, so the two new DNA molecules are genetically different. One daughter cell is genetically the same as the original cell, and the other contains a DNA coat that results from the formation of the capsule around the bacterium, which results in pathogenicity. Transformation can occur naturally or done artificially.